In recent years, it has become increasingly clear that humans have developed an essential symbiotic relationship with microorganisms. Most of us already know the key role microbes in our gut play in protection from pathogenic organisms and the development and maintenance of immune responses, and that changes in our gut microbiota have been linked to obesity and specific cancers. But what about our lungs, the largest interface between the human host and the external environment, being exposed to more than 8,000 liters of inhaled air each day? Although our exposure to microbes and their components is important for the development of a healthy immune system, what happens when we're in an urban environment, where we're exposed to much more than just airborne microbes, but air pollutants too? We all know about air pollution. It's something that affects us all. We've seen it in the papers. We know that it not only leads to health effects, but it decreases our quality of life. But air pollution is a very abstract term. We have to ask ourselves, well, what is it that leads to these health effects? The World Health Organization says that it is particulate matter, or PM. But what is particulate matter? Particulate matter is a heterogeneous combination of solids and liquids suspended in the air, and typically small enough to be respired. It can be made up of reactive gases, organic species, ions, salts and minerals, transition metals, and finally particles of biological origin, such as bacteria and their components. The next question we have to ask ourselves is, how does particulate matter cause the health effects? Research tells us that PM primarily affects the respiratory system, causing inflammation and oxidative stress. What we don't exactly know is which components of PM are the main drivers of this reaction and what are the mechanisms involved. Of these components, bacteria, or more specifically gram-negative bacteria, are well known for their high pro-inflammatory capacity via lipopolysaccharides, also known as endotoxins, found in the outer cell walls. Endotoxins have mostly been studied in an agricultural environment, where they are expected to be in exceptionally high concentrations due to multiple sources. However, to attribute their role in PM-related inflammation, we need to know if endotoxins are even present in an urban environment. Several studies from around the world have monitored these urban airborne endotoxins. However, they reported very low values, thus discouraging the importance of ambient endotoxins in an urban environment. When we investigated the methods used in these studies, we saw that they all relied on filter-based collection strategies, which are typically used to collect PM. However, the problem with these methods is that once the endotoxins are captured onto the filter, they're very difficult to remove because of their lipid portion, and thus cannot be analyzed. Improving on these methods, our study used an impinger-based sampling strategy, depositing the endotoxins directly into water and obtaining more than 30-fold higher concentrations. So now that we know that endotoxins are indeed in significant concentrations within an urban environment, it urges us to reevaluate their role in PM-related inflammation. To do this, we will investigate whether samples with higher endotoxin concentrations indeed induce the highest inflammatory response, what the contribution of endotoxins are compared to other PM components, whether there are any synergistic effects between the other pollutants, and whether we can gain mechanistic insight into PM-related inflammation through various signaling pathways, all of which will be discussed in Part 2. See you then!